This is an enormously exciting occasion here at IPFW to kick off this year's class of IPFW's featured faculty. My name is Bill McKinney. I'm the Vice Chancellor for Academic Affairs. And our featured faculty program gives us an opportunity on an annual basis to really focus in and highlight the outstanding research being done by 4 of our many outstanding faculty here at IPFW. And so this is the kickoff of the lecture series for this year. Yesterday we introduced all 4 of the members of the 2011 class of the IPFW featured faculty. And so without further ado, to introduce today's speaker, I give you Professor Kingsbury from the Biology Department. Bruce?
Well, thank you. It's my pleasure to introduce Dr. William DeMott today, not only as the first of the featured faculty, but he's also the outstanding recipient of the Outstanding Research Award this year, so kudos for that as well. Bill's going to tell you about his work and a little bit about his history, and so I'm not going to say anything to that effect at the moment. The thing that I thought I would focus on was his being a colleague of mine in the biology department, and to start off with, when I came here nearly 2 decades ago at this point, he was already well established and a model researcher as far as I was concerned. He published in excellent journals. He was a recipient of NSF, or National Science Foundation awards, which are hard to come by anywhere, and particularly a challenge at an institution like ours. And so that's very noteworthy. And he also did something which he's no doubt going to talk about a lot today, which is he traveled widely and went to prestigious locations to work with his international colleagues. He's a keen scientist in other ways. He gets very focused on what he's doing at the particular time. And as somebody who studies snakes, that's me, I appreciate somebody who studies flasks of green water, as you'll see. But he has a good reason for doing that. He uses systems that allow him to work effectively to look at big ecological questions, and you can't really work on tuna the same way that you can work on plankton. So it makes a lot of sense. And last but not least, I wanted to say that Bill is also a good departmental citizen and a valued teacher, and I appreciate those sides of his persona as well. And without further ado, please welcome Dr. DeMott.
So my title is Life, Death, and Evolution in Plankton Food Chains, and I just want to start off to make sure that everyone knows what we mean by the plankton. So the plankton are small animals and microscopic algae that are very abundant in lakes and oceans around the world and form a very important base of the food chain. The animals are called zooplankton, and most of them are crustaceans. That means they're related to shrimp and crabs. And one of the key species, one of the key groups in my talk today is this one here called Daphnia. Now I actually brought the clone that I've done my research with me. It's in this little jar, and if you come up later on, you could take a look at it. This is a really big species, so if I had students working and they got some Daphnia this big in their net, they'd say, wow, Dr. DeMott, these Daphnia are really big. They're almost 3 millimeters long, which means about 1/10 of an inch. Some of the zooplankton are insects, so here's one that's an insect. And the copepods, which are important in both lakes and oceans, are probably the most abundant multicellular organism on the earth because they're so important in the oceans. Now, my research really can be talked about in 2 ways. Sometimes I describe myself as a limnologist, That's a scientist who studies lakes. And we limnologists get together with the oceanographers. So we have a society called the American Society of Limnology and Oceanography, also known as ASLO. And in a little over a week, I'm going to be going to the annual ASLO meeting. Fortunately, it's in Puerto Rico, which should be pretty nice. And I'm going to be talking about some of the same work that I'm going to be talking about today. So you're getting a little bit of a preview of that. And I'm pretty excited because I think some of my results are going to surprise and maybe even dismay a few of my colleagues who will be there. I teach a class in aquatic biology, and I'll be teaching that the next time in next fall. The other area, I describe myself as an ecologist, which is the study of the interactions between organisms, between different species, and between species and their environment. One of the cases I want to make, and Bruce mentioned it, is that the plankton are a really good system for studying ecology. Basically, life is fast and dangerous in the plankton. You can have death rates in the zooplankton of 30% a day. And so lots of things are happening there. Generation times are only on the order of a week for zooplankton, on the order of a day for phytoplankton. And this means that since evolution is measured in terms of generations, evolution also happens really quickly in the plankton. And so I teach two classes in ecology at IPFW. Quite a few students here have been in those. One is intermediate ecology, and the other is an advanced class in population ecology that I'm going to teach right after this lecture. So here's an outline of my talk. I want to teach to you a little bit about ecological food chains, and especially talk about defenses. I'm going to tell you a little bit about my travels around the world and my current research, and actually my research for the last 10 years or more has focused on what I'll call the food quality controversy. And part of that has been a collaboration with Alan Tessier, who was at Michigan State, so I spent a lot of time working with him. And I'm also going to be talking about some work that I did at the Netherlands Institute of Ecology when I was on sabbatical in 2009. And in fact, that's the project I'm going to talk about for my meeting in San Juan. If there's enough time, I'm going to go a little bit one step beyond what I'm going to talk about in San Juan. I'm going to say, well, what are the interesting new questions that arise from this work that I could then test, say, beginning this summer. So here's some basic ideas about food chains. One is that most species are both predators and prey. So if you're going to survive, you have to both have defenses against eating, but you also have to overcome defenses of your prey. Another aspect, a basic principle in economics, is they're both costs and benefits. So having too much defense is detrimental when the risk is low. Another thing in ecology is that different predators require different defenses. So a defense that would be effective against one predator might actually be detrimental against another. And then something I'll define in the next slide, and that is the idea that indirect effects are important. So here's a basic pelagic or open water food chain. This one would be one for the Midwestern North America, but it's essentially the same as what you would find in oceans as well. So we have at the top fish that eat other fish. Then we have smaller fish that eat plankton. And among the plankton, some of them are predators, shown on the left, and some of them are grazers that eat the algae, and then the algae are dependent upon nutrients. Now the arrows in this figure represent the direct effects, the direct consumption of one organism by another. So for example, the fish eat the grazers. But the fish also have an indirect effect on the grazers because they also consume these predators of the grazers. So this is a positive effect here. That's a negative effect there. In fact, one of the lakes I studied in Italy, the indirect effect was much stronger than the direct effect, such that fish predation on the invertebrates had a bigger effect than fish predation on the grazers, even though the grazers were the main food of the fish. Now another important indirect effect is going to be the effect of fish on the algae. And basically that's going to be the a main theme in my talk today, because I'm mostly going to be talking about this part of the food chain. Okay, I wanted to say just a little bit, though, about the fish invertebrate predator kind of combination. If you want to avoid being eaten by a fish, fish are looking for their prey, finding them individually. So the best way to avoid being eaten by a fish is to be very small, to be transparent, and during the day to go deep into the lake or the ocean where the fish can't see you, and then you can come up to the surface to feed at night. On the other hand, if you want to avoid being eaten by an invertebrate predator like the fearsome phantom midge, the phantom midge actually locates its prey by the swimming vibrations. So it doesn't look for them, it doesn't use its eyes, and it's small relative to the size of the prey. So if you want to escape from the phantom midge, it helps to be bigger. It helps to develop spines and bigger heads, so you're harder to swallow. And actually, one of the best escape responses is to stop swimming. So then the predator won't know where you are if it misses you on the first strike. Okay, this writing might be a little bit fine for those of you in the back, but I just wanted to say something about my experience as traveling around the world to study plankton. Plankton are found in all the lakes all around the world, many of the same species, so that works pretty well. I got an undergraduate degree in biology. Then I traveled a little bit under the auspices of the United States government, not to study plankton. Then I got my master's degree and PhD degree. And before I came to IPFW, I was a postdoc, means a temporary position, at the Max Planck Institute für Limnologie in northern Germany, which is really the premier institute in this field. And so this gave me a really running start when I got to IPFW, because I had lots of publications, ideas for new grants, and new research. The first part of my career, I mostly worked at Crooked Lake in northern Indiana. Then I took a sabbatical, 2 summers and 1 academic year in Germany. I worked really hard on research, but I also traveled all around Europe, from England to Netherlands to Moscow, giving talks about my research, which was a lot of fun. I then spent several summers at the Center for Limnology in the Netherlands and started doing a collaboration at Michigan State at a place called Kellogg Biological Station, which is some land donated by William Kellogg of cornflakes fame, but it's a Michigan State lab. And there were actually 6 summers when I supported by NSF where my students and I went up to Michigan more or less for the whole summer to do research. And most recently then, I've had a 6-month sabbatical at the Dutch Institute of Ecology, and I also spent some time at the Italian Institute of Ecosystem Studies. So I started out with a small grant from IPFW, then I've gotten a lot of grants from the National Science Foundation. I've been able to pay students to work with me, which is, I think, a good deal for them. I've published quite a few papers, and I've also worked really hard as a reviewer or editor of scientific articles. So I have a lot of experience. Practically everybody in my field, I've reviewed one of their papers. So that means wherever I go, I always see people who know me in that context. So I just want to show you a little bit. This is the town, my hometown in Germany. It's called Plön. It's in northern Germany, and the institute is there. You can see it looks like a pretty good place for studying lakes. It has what's known as the prettiest train station in Germany, right there. Here are some of the people I worked with. Ralph studied the defenses against Chiabris. Dirta got her PhD studying about fatty acids, and we actually published a paper together. The director of the institute right here is Winfried Lampert. So he's one of the world's foremost limnologists, and he was the one who invited me there. He's now retired from being head of the institute, and he's taken a position as a professor at the University of Bergen in Norway, just as sort of a retirement job. On the right is Irina, who's a real good friend. She's from Belarus. She's a scientist, and she's actually now been able to immigrate to America and get a job in, at the University of New Hampshire. Okay, this is the prettiest lake I've worked on. It's Lake Maggiore. This is actually a picture of only one bay of the lake. In the background is the Swiss Alps, and it's in both places. My wife and I came to this lake by train on a stormy, dark night, so I didn't get to see the lake. But then I got up in the morning, opened the door of the balcony of the hotel I was staying at, and took this picture, which was really pretty moving to me. This is a real mecca for limnologists. If you can read this at the bottom, it says Richard Vollenweider, limnologue, and it gives his years. So they actually name the streets after dead limnologists in this town. Now, I've never been able to take any IPFW students with me to Europe, only to Michigan. If some of you are going to be graduate students, though, you might want to follow the, what Kristen did here. So here's Marina, who is my Italian colleague, and she's talking to Kristen, who's actually a graduate student from the University of Vermont. And Kristen somehow told her academic advisor, you know, I'd like to go to Italy to learn some lab techniques. Could I go to Italy for a couple months? And that's a pretty nice thing to do as a graduate student, so that you might want to consider doing that if you can. Okay, so here's what I've been working on. And again, I call it the food quality controversy. This is the idea that often the zooplankton grazers have plenty of food, but they don't grow very well because the food is not that good. It's either deficient in something or it's difficult to digest. And so the three main hypotheses have been not enough phosphorus, not enough of certain fatty acids, and poor digestibility. Now, I've published papers on all three of these, and all three of them are certainly happening some, but it's still a pretty big controversy as to which one is more important in which situation. And in fact, most scientists are kind of on board for number 1 and number 2. And when I go to give my talk, I want to say, well, sorry, but I think number 3 is the more important hypothesis. And this is the idea, well, which I'll be talking about more today, that one defense the algae can have is to survive being eaten, go through the gut, and still live to reproduce and survive. So it's not a 100% possibility, and certainly I actually have done a lot of studies on what favors survival and what leads to mortality. But it's a kind of interesting defense, being eaten but surviving the fact. So I want to give you a little background on the phosphorus. This is a more elegant theory. Phosphorus is important for all living things, and you probably know that many times phosphorus is the limiting nutrient for algae in lakes. Not enough phosphorus, so the algae doesn't grow very well. When the algae is limiting, its content of phosphorus is often very low, and Daphnia also needs phosphorus in order to make RNA, in order to make protein, in order to grow. And so the idea of this theory is if the stoichiometry of carbon to phosphorus is greater than 300, probably the algae will not have enough phosphorus for Daphne to grow very well, even if there's plenty of food available. And one interesting thing about the animals that are growing under low phosphorus conditions is this. They grow very poorly, but they get fat. So they have plenty of energy, but not phosphorus. They don't grow for that reason. So I'm going to be showing you some data. And scientists really like numbers and data and graphs. I hope you'll understand some of them, but I want to say something about a lot of the results I'll be showing are exponential rates of change, which is basically saying what is the percent change per time, or in the case of what's happening in a lake, say what percent of the light is taken away for each meter as you go down with depth. And it's also important for economics and that area, so I'm sure that the economists and business people around you understand the importance of compound interest, which is basically what we're talking about. And the take-home message is that sometimes what look like fairly small changes in exponential rates are really big if they occur over a period of time. Okay, so I'm going to tell you about some of the methods we use. And in many of my studies, one of the key methods is called the Daphnia juvenile growth bioassay. Now, Daphne reproduces asexually so that females produce eggs that don't need to be fertilized, that are genetically identical to the mother. And this is really nice for experiments because all the individuals in our experiment might be genetically the same. And what we do then is to feed the mothers well, collect a lot of babies that were born at the same time, and we want to test how well they grow with different diets or different food conditions. And what we do is take some of the babies and dry them and weigh them. Then we allow the Daphnia to grow for 4 days, changing the food every day. At the end, we dry them and weigh them. Now, the one thing you need is a good scale to do this. And I have one at IPFW. They have good ones in Germany and the Netherlands. The reason is it's a little bit like weighing pieces of dandruff. And in fact, everyone knows what an aspirin is. An aspirin is 250 milligrams, which is 250,000 micrograms. So my scale is such that you could break your aspirin into 250,000 pieces and weigh those pieces. Okay? So you need a really good scale for this. And then you can calculate an exponential growth rate, which is basically what is the percent growth per day compounded instantaneously. So let me say a little bit about this collaboration between me and Alan Tessier. I've been doing these studies mostly in the lab, some in IPFW, some in Germany, some in the Netherlands, studying these different mechanisms of food limitation under very controlled lab conditions. Alan had been studying 30 lakes in southern Michigan, and he found very consistent differences between lakes in the quality of the food. And so we got together and said, let's use DeMott's lab techniques, combine them with Alan's lake to try to understand what's happening in those lakes that is leading to consistent differences between lakes in food quality. So my students and I spent, I guess it was 5 summers working in Alan's lab, and we had good grant support to do that. On the left is Alan. We're about the same age and area of our career. We both can be obsessed about plankton. That means we sometimes dream about it. When we're eating our food, we're thinking about experiments. When we're exercising, we wonder, how can I solve this problem? At the right is Pam, who's Alan's technician. She's a master's level person who helps run the lab, runs the experiments. And she was a great mentor to the IPFW students. So she looked after them, taught them how to do the lab projects, and so forth. So this is an overview of what Alan found. He found big differences between lakes that divided into 3 categories of depth. The deep lakes are called deep refuge lakes, so they're deeper, and they have oxygen in the deep waters. And in those lakes, Daphnia can migrate into the deep water during the day and come up to the top at night to feed and basically avoid risk of being eaten by fish. So in those lakes, we have large numbers of big Daphnia. In the shallow lakes, have the same fish, largemouth bass, bluegills, but there's no place for Daphnia to hide, and the big Daphnia can't survive at all. The predation is much higher. They might be suffering mortality of 20 or 30% per day by fish. And the food is much better. We didn't study later the very smallest lakes, but these are lakes that are so small that bass die off when there's snow on the lake in the winter. And these have very small fish. No Daphnia can survive. There's lots of plankton, but the fish that eat plankton get such small bites that they can eat tens of thousands of plankton per day and not really grow very well. So there's a change between more predation going from the bottom to the top and worst food conditions going from the top toward the bottom. I think that ecologists were pretty much expecting this general result, but Alan and everybody was surprised at how strong and consistent this result was. On the top graph from Alan's paper, the deep lakes, the shallow lakes, the temporary lakes, he used the growth assay by taking water from each lake, bringing it back to the lab, and measuring the growth of the Daphnia, asking the Daphnia, how good is this food? Each data point represents 1 year's data from one lake. So this is like 30 or 40 lake years of data. And in the deep lakes, it was consistently yucky food. In the shallow lakes, the food was very good. And in the really shallow lakes, the food was phenomenal. Now, Alan then looked to say what correlates with these food differences. And basically what he found was it had nothing to do with the amount of food. And so when he measured food by, this is phosphorus, this is chlorophyll, this is carbon. These are the deep lakes, these are the shallow lakes, and those are the very shallow lakes. There was no relation between the amount of food and how they grew. So the idea seems to be food quality. And Alan, based on the kind of algae in these lakes, thought it might be the digestion-resistant situation. So this is our plan. We got funding to do this. And sometimes you do experiments and you say, well, I wish I would have done it a little bit differently. Sometimes you do experiments and, wow, I really hit that on the nail in terms of how to do the experiment. And fortunately, this one was a really good one. And the one I'm going to talk to you about from the Netherlands were experiments where I was really happy with how it was designed. Basically, we took 3 of the shallow lakes, 3 of the deep lakes. We did experiments in each lake twice. And we did growth assays where we added supplements to see how the animals responded to the supplements. And then I also developed a new technique for labeling the natural algae with radioactive phosphorus to test its digestibility. So those are 2 of our lakes. And it was really nice in the summer in Michigan to get up early in the morning, go out to these lakes, and collect some water, usually 2 lakes per week. And then we came back, and that shows me I've got all these little jars, and I've got Daphne in it. Every day I have to change the water, add new supplements. And it took about 4 hours a day, 4 days a week, 6 weeks, or I guess it would be 4 weeks during the summer. So it looks like it's pretty tedious, but if you're getting good results and you're really wanting to know what happened, and it's pretty exciting, then it's fun. Back there is Megan, who is now she was a graduate student, now she's a professor at Georgia Tech. So this is our first results. This is the controls. This is how well the Daphnia grew in the water from the lake, where we took the water, brought it back to the lab, put the Daphnia in the jars, put that in a chamber that kept it at 20 degrees Celsius, 68 Fahrenheit. And as expected from Alan's work, the growth rate here was higher in the shallow lakes than the deep lakes. It was unrelated to the food concentration. So the open circles are deep, solid ones are shallow. Each data point's the mean for 1 week's experiment from one lake. On the right, you can see the C to P ratio. That's the number of carbons per phosphorus molecule. And in fact, this shows the pattern expected for phosphorus limitation. If the animals are growing better when the phosphorus ratio is lower, when there's more phosphorus in there. But that doesn't really prove what's happening. And so this shows a big experiment, and it was possible to present the whole experiment in one graph. So that's a nice thing to do when you're publishing. Basically, we looked at what we call a growth differential. So we have the controls I just showed you. How well do they grow in the lake water? And then we have different supplements we add in. And because you can subtract exponents, we can take the supplement growth rate, subtract the control, and get a measure how much did that supplement improve growth. So this graph shows for the deep lakes and the shallow lakes, for 3 different lakes, and for 6 experiments per lake, shows the mean and standard error. If we doubled the food concentration by centrifuging, it improved growth a little. If we added essential fatty acids, it improved growth a little. If we added phosphate, hardly anything. If we added algae that was deficient in phosphorus, helped some. If we gave them algae that was very easy to digest and rich in phosphorus, they just took off and grew like crazy. Now, for those of you in the back, I don't know if you can see this, but I just want to show you an idea of what these growth rates translate into in terms of how much the animals are growing. So these baby Daphne usually weigh about 5 micrograms each, where a microgram is a millionth of a gram, and there's 455 grams per pound, so it's not very much. In the shallow lakes, they grow from 5 to 17 in 4 days. If you add phosphorus, they grow to 18, but if you add the Stoichiometric, they grow to 33. In the deep lakes, they grow from 5 to 8 in 4 days. If you add the phosphorus, it's from 8.4 to 8.8. But if you give them Stoichiometric, they've tripled their weight by adding this supplement. And so what this suggests is that they're not getting enough energy from the algae in the lake, but doubling the amount of food in the lake doesn't really do anything. So this suggests that it's probably digestibility and not the amount of food that's leading to this low energy. So the last thing we did was a kind of a new technique that I worked out. I didn't know if it would work, but it worked pretty well, where I labeled the natural food with radioactive phosphorus to try to measure how well they could digest it. And so the x-axis here is a measure of digestibility. The y-axis is the growth assay data. And then these are experiments from the 3 deep lakes and the 3 shallow lakes. Shows that digestibility seems to be the important thing that differentiates these kinds of lakes. Moreover, the deep lakes have a lot of algae that can survive being eaten. And so this is actually a part of the Daphnia gut. And these are algae that are pretty much intact, and they have different coatings on them which can allow them to survive being eaten, at least under some circumstances. It depends. So if the food is low, it stays in the gut longer, and it's more likely to be digested. But there's certainly a lot of survival. Okay, I just want to show you a couple of pictures. We also did some large enclosure studies. So these are 3,000-liter bags, where a liter is about a quart. And this is Allison, who was one of the IPFW students. This is Klaas, who was a student I met as a PhD student in Germany. And then we hired him to come to America. He's actually Swedish, but he got his PhD in Germany. I also had 3 students who are co-authors with me on some of these studies. These were all important papers. All 3 students got honors degrees in biology based on the research they did with me. And then Bryn and Aaron doesn't like to be photographed, also got the Outstanding Biology Senior Award. And Bryn and Jenny both gave talks like I'm giving today at national meetings, one in Santa Fe and one in British Columbia. So those were pretty good experiences for them. So now I'm going to get to the last part of my talk. This is the work I did in the Netherlands. This is what I'm going to talk about at the Oslo meeting. My title is A Test of the Effectiveness of Algal Defenses in Phosphorus-Limited Microcosms. Basically, a lot of people, the work we did in Michigan was published. People said it was really good. But then people in other places said, are those defenses really that strong? Are those Michigan lakes typical, or maybe the lakes I'm studying in Colorado or Norway or Germany are different? So you want to have more ways of studying this. And I wanted to then use a more controlled laboratory system, which are microcosms. Ellen von Donk is my main collaborator. She's chair of the department at the Institute. If you go visit there, they give you a mansion to stay in. except you only get a small apartment on the top floor. You don't get the whole building. And it's on the Vecht River, which flows through northern Netherlands in between Amsterdam and Utrecht. If you go further down the river, there are lots of beautiful houses on the river, and one of them is this castle that was built during the 12th, I guess the 13th century. So this is the system I studied. I did experiments in these 10-liter carboys. And so actually when I was in the Netherlands, I had my own room. I shut the door, didn't see anybody, although I didn't stay there all the time, but I stayed there a lot of the time. And this is my experimental design. I did experiments with 18 of these 10-liter carboys. So there's 3 replicates, carboys that are the same. Half of them got Daphnia, half of them had no Daphnia. Some of them used an undefended algae, some used the defended Oocystis, and some were a mixture. And I ran this experiment under conditions where I thought phosphorus would be very low. Now, other people have done this experiment, but they only used this part of it. They didn't use either of these. Here's a picture of my Enkystrodesmus, which is actually growing here. The Daphnia that's up there is actually the same, genetically the same as the one here. And not too long ago, I got a message from a student who is now a PhD student in Quebec, but actually is from the University of Warsaw in Poland. And he said he took this clone, brought her with him to Warsaw, and she participated in a research project that's already been published from that study. This Daphnia here has been feeding on Enkystrodesmus, and its gut is kind of brown toward the end, which means the Enkystrodesmus is well digested. This is a close-up of this same area here. I don't know if you can see it, but there's intact cells here that are not digested, which is an Oocystis. And actually, the Oocystis I used in this experiment I got from Crooked Lake at Columbia City and brought it with me to Netherlands. So I'd have a good algae to work with in this study. So some predictions. If the Oocystis is really well defended and it's the only food, we expect the Daphnia is not going to grow very well. Another prediction is if the Oocystis is well defended, it should survive when the predator, that is Daphnia, is abundant. And if there's a cost to this defense, one possibility is that the Oocystis will not grow well when there's no predator present relative to an undefended algae. And particularly, you might expect that the undefended algae will be able to grow faster in the absence of the predator than can the defended algae. So that would be a cost of defense. Okay, this is meant to give you a little bit of an idea. I'll be a lot more technical when I give the presentation in San Juan, but this tells what different people are doing. So Ellen's main job was getting the money and helping with the overall plan. This is my wife. She's a teacher, and she was able to come along on sabbatical by taking a semester off with no pay. And I told Peg, if you work in the lab doing some of the weighing for me, we'll have more time to take some trips on the weekend. Oops, 2 people here. Suzanne and Nico are both master's level technicians, so they have full-time jobs working at the lab. Nico did the automatic carbon analysis and phosphorus analysis. Suzanne took several weeks to count the algae. And this is a picture of me in one of the rooms where we're filtering water, something limnologists do a lot of. And we have really nice equipment, very expensive microscopes, and other things. And so we did get to go to other places. Peg and I often travel on Big Red. That's this bicycle here. And Big Red comes apart and fits into a suitcase. So we brought him with us to the Netherlands. We brought him with us to Italy, not to England. And the cool thing you can do in Netherlands with a bike is to decide you're going to go visit some famous city. You get on the train with your bicycle. You go to that city. You spend several hours walking around the city. And then you want to calculate which way the wind's blowing as to which city you go to. Then you can ride the bike back home. And so typically we'd go 30 miles, 50 miles, 80 miles, go visit a city, and then ride the bike back home. It works pretty well in the spring and summer, not so well in the winter when it gets dark at about 3:30. So that doesn't. So anyway, we did a lot of cycling as well. So this is an initial experiment I did, a growth assay with the algae I was using as an experiment, with the algae that are growing under good conditions in the lab. And the direction, so this is the growth assay. That's the, you need to remember, Enkystrodesmus is undefended, Oocystis is defended, and there's the mixture. So that makes sense, but actually I was a bit disappointed that the defense wasn't stronger, and I was worried about that at the time. This shows what those kind of exponential rates translate into over 4 days. So this says that a 5 microgram baby becomes 53 in this food, 30 in this food, and 20 in this food in 4 days. So it's definitely well enough to measure it, and that's highly statistically significant. Okay, this shows the main results of the experiment with respect to the zooplankton. Each data point is the mean and standard error for 3 different enclosures, or 3 different mesocosms that were replicates. And what we see is that this is the Daphnia abundance. They grew in the beginning, but then with the defended algae, the population crashed down to very low levels. Now this is a log scale, so this goes 1 to 10 to 100. And so these are really big differences in the amount. The growth slowed and then slightly declined with the undefended and with the mixture. We can also see big effects in the clutch sizes. So the clutch size is how many eggs does an adult female produce? And by the first sampling, they're producing 10 eggs with the mixture and undefended, but only 2 eggs with the Oocystis, and that quickly went to 0. Another kind of data I looked at was the size of the animals, which ones are adults, which ones are juveniles. And what you can see is that early in the middle of the experiment, in the Oocystis, it went from a lot of juveniles to almost all adults. And basically that was because all of the juveniles starved to death, and adults tend to survive under starvation better than juveniles. Okay, this is a little bit complicated, and I don't want to go over all this data, but these are some of the carbon and phosphorus data from NICO. And so I have the open circles are going to be the ones with no Daphnia. The closed ones are the ones with Daphnia. The circles are the undefended, the squares are the Oocystis defended, and the triangles are the mixture. But what I want to show you is the C to P ratio. And basically, if it gets above 300, there's not enough phosphorus. And you can see that by day 6, all of the microcosms with Daphnia were at about 1,000. So what I did then were some of these growth assays where I actually took water from the microcosms, grew the Daphnia in it, and then grew them in that water plus phosphorus to try to understand what was happening there. And I did a lot of different growth assays, but I'm only going to show you this one because we're going to run out of time, and it gets a little bit complicated. Okay, these were the 3 for the lab culture I just showed you. By day 6, the food was pretty bad, and in fact, for the Oocystis, the animals were actually losing weight. This is pretty unusual for these animals to lose weight. And in fact, this was happening when the animals were dying and starving. So this really makes sense that the food was really bad. If we look up here, we see the abundance of food, which actually increased quite a bit. So this decrease in growth is not due to not enough food. There's really way more food than they need. But the C to P ratio, the number of carbons for each phosphorus atom, has gone way up. So it could be that the food is bad because there's not enough phosphorus. Now, in fact, then I have the next experiment where I added the phosphorus back in. And what you can see is that for the Enkystrodesmus, it went from here to here, almost up to here by adding phosphorus. For the mixture, it also improved. So in fact, those microcosms, the animals were not growing because they didn't have enough phosphorus. On the other hand, for the Oocystis, there's a slight improvement that's not statistically significant, and the food is still terrible even if you add the phosphorus back in. So the conclusion from this is that actually that the defenses against digestion are greatly enhanced under conditions of low phosphorus. They develop a stronger coating to protect themselves from digestion. And so the other thing that's really interesting to look at here, if you looked at these animals, the animals here and here are really fat. Now that doesn't mean that they have big bellies. It means that if you look at them, you see all these little fat droplets. And so they were maxed out on fat, whereas the animals here had zero fat. So they had no energy left, and they were starving to death. Okay, here are Suzanne's algal counts. Notice these are also a log scale. And this is going from 500 algae, this is thousands per milliliter. So it's going from 500 algae up to almost 200,000 per milliliter. And a milliliter is 20 drops. So they can have a lot of algae in there. And again, the dark circles are the dark symbols are the ones with Daphnia. The open ones are without Daphnia. And the one that really tells is when they're in a mixture. So those algae in the mixture are being preyed upon by exactly the same Daphnia. And what you can see is this is the Oocystis without Daphnia. This is the Oocystis with Daphnia. Hardly any effect. This is the Enkystrodesmus without Daphnia. This is the Enkystrodesmus with Daphnia. This is 500, and this is 150,000. So there's a tremendous defense there that works for the Oocystis in contrast to what's happening with the Enkystrodesmus. So I actually wanted one of the last things I want to show you is what this actually looks like if you collected the samples. So this is early in the experiment, before the things got too extreme. But on the left are two of the Enkystrodesmus microcosms with Daphnia. The Daphnia have been filtered out, and then I'm going to process this and measure what's happening, measure the amount of carbon, phosphorus, the number of algal cells, and measure the volume of particles. So those are pretty clear. This is without the Daphnia. So you can see that, and that's only halfway through when the differences aren't too big. And this is what it looks like for the defended algae. They didn't grow as well in any of them, but this difference is actually significant between the grazed and the ungrazed, but it's certainly a lot less dramatic when you have strong defenses there. Okay, one of the last things I want to show you is, can we detect differences in the growth rates of the algae? And one kind of data I collected was particle volume. And I used a particle counter. I have a different model of this in my lab. And basically this little machine, they use it in hospitals for counting and measuring blood. But you can put a sample of algae in, and it can count 10,000 algae in 1 minute. It can measure them all, how big they are. It doesn't tell you what their species is. Looks like something you could buy in Walmart. However, it costs about $50,000, so you probably not what your average consumer would want to work with. But what we're looking at here is what is the slope of this growth curve early in the experiment when the algae are growing exponentially and in the enclosures, microcosms without the Daphnia. And if you do a little regression where you plot time on the x-axis and the natural log on the y-axis, the exponential growth rate is shown here. This is the ones with the Enkystrodesmus. This is the ones with the Oocystis. Now, it's the slopes, not the positions, that are important there. And if you could read that, the slope of this one is about 0.9. The slope of this one's about 0.6. And so this shows the differences in the maximal growth rates. Just to emphasize that exponential rates really make big differences, This gives a doubling time for the population of about 18 hours. This gives a doubling time of the population for about 26 hours. And if you started with one cell at day 1, after 1 week, this population would be 672, and this population would be 85. So that difference between 0.9 and 0.6 in a few days makes a really big difference. So here's my summary and conclusions. I'm not really quite satisfied about this for my talk in the Netherlands, but in my experiment here, all of the algae were phosphorus limited. The Daphnia got phosphorus limited when they were growing on the Enkystrodesmus or mixed. But the thing that I didn't really expect, although there's some evidence that this could be happening, is that when the digestion-resistant algae are grown under phosphorus-limited conditions, their defenses are strongly enhanced. So the experiment that I did first with the good lab algae really underestimated the strength of the defenses under conditions of strong P limitation, which is also the way it was in those Michigan lakes that Alan and I studied. The deep lakes were strongly phosphorus deficient. And so this helps explain why the digestion defenses were so strong. There also seems to be a trade-off between growth rate and defense. So if you don't have a defense, you can probably grow faster. That means if there's no one around to eat you, you might have an advantage if you don't have the defense. The people who do the stoichiometric theory, and I'll be seeing some of them in San Juan, they like to think that you can just measure that C to P ratio and predict what's going to happen. And the problem is if there are any defended algae, that won't work. So I don't know if I'll go through all this, but I think the next thing to do would be to go back to some of these lakes and isolate some of the algae. And certainly we need to do some more testing to see whether what I saw in the Netherlands is it just from that one strain of algae, or is that a general characteristic? Something I think will be really interesting to see is whether the algae evolve defenses proportional to the kind of grazing they experience in a lake. In other words, is there evolution occurring at lakes such that the algae in a particular lake are fine-tuned to that lake? We know that deep lakes have more digestion-resistant algae. Shallow lakes have fewer digestion-resistant algae. But I'd like to know whether the digestion-resistant algae in the deep lakes are in fact more digestion-resistant than the ones in the shallow lakes. So that's some research that I hope to start this summer.